tumor incidence is known to increase with age. However, tumor progression often proceeds at a slower rate in aged organisms. It is known that many human cancers, although not all of them, evolve at a slower rate in old than in young individuals. Here you have a partial a list of those tumors which behave in this way. And you can see that these are the tumors which are the most frequent in humans. We were interested in the mechanisms responsible for this age-related decreased malignancy of tumors. What happened? Okay. We used two murine models, B16. What happened? No, but it was okay. We used two murine models, the B16 melanoma and the ATR lymphoma. You can see that tumors from old mice are much smaller than those of young ones, and the same was we observed in ATR lymphoma. We measured this difference also by, sorry. Excuse me. By measuring the size of the tumors, and you can see that the black which are the tumors in old mice are smaller than those in young ones. This is also expressed by survival of mice. We tested the possibility of several mechanisms. Some were tested by other groups, and some only by ourselves. We then tested whether there is a difference in proliferative capacity, in apoptotic cell death, in angiogenic ability, immune response, and tumor cell ploidy. We first then tested for cell proliferation and apoptosis. You can see that apoptosis is much more evident, according to apoptag, in tumors from old than in those from young mice. Yes, pardon. Yeah. Here you can see, according to flow cytometry, DNA flow cytometry, both differences. You can see that the G2M fraction is larger here in tumors from young mice than in those from old. But on the contrary, the fraction of apoptotic cells is larger in old than in young mice, and this is seen as well quantitatively. You can see the difference in apoptosis according to according to the typical DNA fragmentation, ladder type, that it is more evident in old than in young, for B16 melanoma and for ATR lymphoma. At the molecular level, we can see the evidence that apoptosis is stronger in old than in mice, since we have lower BCL2 expression in old mice in B16 melanoma than in young ones. In ATR, this can be seen the same, but FAS protein is higher in old than in young mice, at least in the ATR lymphoma. We then compared angiogenesis according to macrovessel density in tumors from young and old mice. And you can see in the factor VIII staining that in old mice vasculogenesis is more evident in young than in old mice. We then compared the immune response and we look only here, since you have seen already. The macrophage content in old mice is much more evident in tumors from old than from young mice. So we saw until now 4 possible 4 possible mechanisms of this phenomenon, this interesting phenomenon. And then we observed something more that we did not look actually for, but we observed it, and it is a cell ploidy. We saw that the B16 melanoma in young mice are larger size than in old mice, and we tested why. And what you can see is that if you look at these 2 curves, we see here the size of tumors according to FACS data and forwards data, and you can see that tumor from an old mice, in this tumor we have larger cells than those in the old. So it also suggests that these cells are smaller than in old than in young mice. And we see something more. If you look at here is the cells of a tumor of B16 in old mice. We see here peaks of apoptosis, diploid cells, tetraploid cells, and octaploids, or even cells of higher ploidy. While in the young, there is this peak of diploid cells is not evident here, or anyway, there are a few cells. So it seems that in the tumors from young B16 melanoma are tetraploid, which is was in fact observed by other groups as well. And the diploid cells are at a lower proportion in tumors from young. What we see here in the third column is again apoptotic cells, small and with less DNA, low DNA content. This is in fact DNA flow cytometry versus cell size forward theater. So we have these apoptotic cells, diploid cells, tetraploid, I hope and cells of higher ploidy. What we did here, we calculated the various subpopulations of different ploidy, and you can see that diploid cells are a larger proportion of diploid cells is seen in old, the blue, than in cells from young mice. In both B16 melanoma and its variant B16F10. By contrast, the opposite picture is seen with respect to tetraploid cells. We have less tetraploid cells in old than in young, in old than in young, okay? Tetraploidy is considered to precede aneuploidy, which in turn is at the origin of genetic instability in tumors. Aneuploidy is nowadays one of the hallmarks of cancer. It is the most common type of genetic instability in cancer. Now, what we found here, the tetraploid to near euploidy transit in melanoma cells from old mice might constitute one more mechanism by which the genetic instability which is inherent to tumor progression is attenuated in the old. Our findings indicate that the aging microenvironment can actually affect the tumor cell genome or its expression. Tetraploidy slash tetraploids are considered to constitute a metastatic intermediate between diploidy and aneuploidy. Several mechanisms exist which avoid the generation of vital to eliminate tetraploid cells. Cells that do survive illicitly such cells which survive these checkpoints of tetraploidy are prone to chromosomal instability. And lately, an article of Aylon and Moshe Oren, already mentioned today, is named P53, the guardian of ploidy. However, in fact, not only P53 has this role, and the whole series of oncogenes and tumor suppressor genes contribute to the avoidance of tetraploidization. And I'm sure that it is not the whole list that you will see here. This and in the same article of Vitali et al., you have a whole list, among which indeed you have the P53, but he's not alone, it's a whole list. And another list you have here of oncogenes which have the same role. In conclusion, while the first four mechanisms that I mentioned were mainly related to the effect of age on the multiplication of tumor cells, proliferation, apoptosis, and partially, of course, angiogenesis, which also has other effects. In fact, tumor mass. This last mechanism showed a different surprising effect in a way, demonstrating what effect host age may have on genetic stability, which affects, rather than tumor mass, tumor progression. Thank you.